In this video lecture, we're going to go over the second part of chapter 3 in the cell, and we're going to start off by looking at organelles. Okay, so let's do a quick review. Cytosol being all that fluid component of our cytoplasm. Inclusions being our vacuoles or stored nutrients, so we can have lipid droplets or glycogen granules. And just like our body, how we can function, whether we have digestion or respiratory areas, systems, our body can carry out different processes because of our organs that carry out these reactions for us. Now, in a cell, we also have metabolic machineries through these little organs called organelles. And so this entire lecture, we're going to go over the different organelles and then finally conclude with how they come together for protein synthesis. Alright, so here is our cell. We're going to go over a couple of these organelles. So we're going to look at the mitochondrion, which is this structure over here. We have the smooth endoplasmic reticulum and the rough endoplasmic reticulum, so both of these blue structures. But you notice this is smooth because it's nice, clean. We have nothing on top of this organelle. Versus this organelle over here, we have all these red dots, and that's why this structure is going to be the rough ER. And what's studded on top of this rough ER are ribosomes. And so all these red dots are ribosomes, and not only do we find it on our rough ER, but we also find those ribosomes free-floating within our cytosol. Another organelle we have is this Golgi apparatus, which interacts very closely with our rough ER. So once we can create all our proteins here in our rough ER, we can send that over to our Golgi apparatus. We also have peroxisomes. This is for detoxification. And then we have lysosomes digesting worn out material. And then of course, our most important organelle of them all is going to be our nucleus. And if you look at our membrane and outer area of our nucleus, we can notice there's this double layer of membrane. We also have these structures here in orange. These are called nuclear pores. They allow contents in and out of our nucleus. And then finally, you'll note it's this dark purple structure in the middle called the nucleolus. Now for the next couple of slides, as we go through the different organelles and the different functions of each of these organelles, we're going to compare our entire cell to a city. So we're going to say cell city. So we're going to see how all of these interact and how it's comparable to our city, and then hopefully it'll make more sense. So we're going to start off with the most important organelle being the nucleus, and this is considered the control center of the cell. And why is that? Well, before I get into that, let's just think of our nucleus as our state capital. Well, what goes on in our state capital? And the capital, we can design, pass, and enforce rules and laws to run our entire city. So same thing with the nucleus. The nucleus contains all of the genetic material and instructions needed to synthesize proteins and everything else needed by the body. And why is this so important? Any cell whose nucleus or DNA is damaged will automatically undergo cell death. This is how crucial this entire nucleus is. And so so containing all the genetic material in the form of that DNA, okay, so very important. Now, we mentioned we have this nucleolus found with the entire nucleus here in purple, and the function of this is where we synthesize and produce ribosomes. So this is the production of ribosomes. And once these ribosomes are produced, they can exit our nucleus through that nuclear pore, and we can stud them against our rough endoplasmic reticulum, or they can be free-floating here within our cytosol. And the next organelle we have is going to be the rough ER, you see right over here. And the function of this is to make and export proteins. Okay. And so all of these proteins, so here, everything in red, those are these ribosomes. And we'll get into that the function of these ribosomes is to synthesize to produce proteins. And then once they are done synthesized, they're going to enter here into our rough endoplasmic reticulum. And then from here, all these proteins and how this interacts with other organelles is that proteins made in the rough endoplasmic reticulum which we're going to refer to now as RER for short, will be transported in vesicles destined for the Golgi apparatus. And we can think of this rough ER as this freeway. So of course we only have 3 here within Hawaii, but if you go somewhere like LA or major cities, what you'll notice is you'll have lots of freeways interchanging, going to so many different directions. And so if you can imagine, you have a plant, okay, and we're going to create, what are we going to create in this plant? We're going to create, I don't know, MacBooks, because I'm staring at my computer right now. So we're creating all these MacBooks, right? So you can start generating Apple. This is an Apple. Can't draw an apple. Anyway, Apple Store. So going to start generating all these MacBooks. Well, here's a store over here, but we want to ship it to a whole nother area over here. And so this is going to be like a ribosome. So the ribosome is like the store. It's going to produce all of these MacBooks. MacBooks can be considered our protein. And then how do we transport this MacBook from this site to that site? Well, we're going to put it into a car. That car is going to drive on these freeways until finally it gets to our destination. And so, Apple Store can be just like those ribosomes. Ribosomes produce proteins. Here we have a MacBook that we're being produced. We want to ship it somewhere, so we're going to get into something called vesicles. So here is transport vesicles. So our vesicles are going to be our car. And we can move it from one area to another through all these roads, which are those rough endoplasmic reticulum, until we get to our destination. So that's how we have that analogy. Ribosomes, these are the organelles that produce proteins. Okay, and so they're not, here we have small factories that synthesize only one type of thing. These ribosomes are very diverse. So looking at this one ribosome over here, it's not always going to produce Nike tennis shoes, or it's not always going to produce Big Island candy, cookies, cookies. Anyway, so we're not always going to produce just one thing. We can produce instructions. So this one ribosome can produce this, or it can produce this. But you can look at this ribosomes as a factory where you give it instructions, it's going to start producing its product. And so ribosomes, we have 2 areas once again. We have it along the rough ER. And if the ribosomes are found along the rough ER, these proteins will be destined to go to, outside of the cell, be part of the cell membrane, or form organelles, okay, specifically lysosomes. All right. Now, the other location where we have ribosomes are within the cytosol. And any of these proteins that are synthesized within the ribosomes in the cytosol or cytoplasm, these are going to stay within the cell. So proteins synthesized on free floating ribosomes will stay in this cell. All right, two different things. Now, what are these proteins that can stay inside the cell? Well, they can be enzymes. They can catalyze different reactions within the cell. And so there's actually lots of proteins we have within our cell in itself. They can regulate when the cell will continue to grow, when it will die, and so on and so forth. Smooth endoplasmic reticulum. This is our next organelle. And the function of this is for lipid metabolism. And what that means, it includes cholesterol and fat synthesis, as well as breakdown. Right, so remember, metabolism always build up and then break down. And then how this interacts with other organelles? Well, this actually helps maintain the entire surroundings for the other organelles. So how does it do that? Well, it detoxifies drugs and pesticides. Okay, and so if you look at this illustration here, and an area in our city where we're producing oil, so for cooking or oil for frying, ooh, those malasadas, right? Anything like that, that's what we're going to need those oils for. Also, you can think of drug rehabilitation centers on the island, and that will be for the drug detoxification role of Smooth ER. The next organelle we have is the Golgi apparatus. And the function of the Golgi apparatus is it's the sorting center of the cell. And especially it's going to help send off, it's going to help repackage proteins and lipids synthesized by the smooth endoplasmic reticulum and rough endoplasmic reticulum prior to getting sent out of the cell, or if those vacuoles become part of the membrane. Or if it becomes an organelle. So it works really closely to our rough endoplasmic reticulum. For once those proteins are produced by those rough ERs, we can send it over to our Golgi apparatus. Now, how is this just like our postal service? Well, you have all this mail. You want to send it to different areas of the world within the city itself. And so what do the mailman, or what do they do at the post office? Well, they receive, collect, and then they help reorganize and redistribute all that mail to the specific area where it's supposed to go. So that's just the same role as that Golgi apparatus. The next organelle we have is the mitochondria, and we have two illustrations for this. We have a generator and we have a solar panel. And so what do you think the mitochondria's role is? Great. It's the energy source of the cell. And here is really where we generate all the, well, majority, I shouldn't say all, but majority of the ATP, or energy, needed to drive cellular reactions. And so it does generate the most amount of ATP, especially in the presence of oxygen. So the majority of ATP, it will be produced in the presence of oxygen. If we don't have any oxygen, we do not undergo through the energy production in the mitochondria. But mitochondria will produce the most amount of ATP in the presence of oxygen. Next organelle we have is the lysosome. And the lysosome you can think of in our city as garbage trucks because they will collect and break down any garbage within the cell. So here we can get rid of a lot of our debris. Same thing here with our lysosome. And our lysosome contains, it'll break down worn out or digest. Break down, worn out cellular structures. And foreign substances. Okay, and so how does this interact with other organelles? In the event that any of these organelles are worn out, if they're dying or broken, then these lysosomes can fuse with those organelles and then help digest them and break them down. So pretty much the same thing that we wrote in the function. So it can break down worn out structures, and it can also digest pathogens. Alright, next organelle we have are peroxisomes. And peroxisomes, these are important in reducing free radicals. And what free radicals are is basically these ions that don't have matched electrons, and because of that, they're very reactive. They can bind to DNA, they can bind to different things within the cell, and then cause damage to the cell itself, which is why free radicals aren't very good for our cells. And so peroxisomes, what they can do is they can use peroxidase, which is an enzyme that will convert those free radicals to hydrogen peroxide. Now, we really don't want hydrogen peroxide too much here within our cells. That is also, so we can break this down into water, through another enzyme called catalase. So this is enzyme number 2, enzyme number 1. Okay, and so what organs in your body do you think you're going to have a lot of these peroxisomes? What organ do you think is used for detoxification? Not the organelle, but the organ in our body. Good, your liver and your kidneys. So you're going to see lots of peroxisomes, especially in your liver and kidneys. Okay, and so I like to think of peroxisomes as a popo. You know, any free radical you can kind of think of, or anyone trying to create crime, trying to create disorder within the community, they can help regulate these by like taking them down, putting them in jail, or just kind of putting them in place, right? So those are the peroxisomes within our city. Finally, the last structure we're going to be looking at are cytoskeleton. And this isn't an organelle, but it's actually needed, and it's quite important for a cell itself, because as we looked at the analogy of those houses being like a cell, we need to have some sort of structural support and framework. And that's what the cytoskeleton really is. It's the internal framework of the cell that will determine the cell shape, size, so just cell shape and size. Now, other things it can do, so we have 3 different types of cytoskeleton that we, you can briefly read them, but I'm not going to really quiz you on them. Those are going to be intermediate filaments, you have microfilaments, and you have microtubules. Now, you don't have to know the specific function of each of these for an exam. You might have to know it for one of your quizzes. However, collectively, what these do not only help determine cell shape, cell size, but also the ability for the cell to move itself. So, movement of cells. And that really has to deal with some of these, but not all of these cytoskeletal filaments. Okay. Now here is a video you can watch on your own time, and this video, I think I might have uploaded a different one for you, but this is a great way to help you review the different organelles found within the cells and how they all interact with each other. Okay. Now this last part of lecture, we're going to go over the nucleus of the cell. And why is it so important? Because once again, it contains all of the genetic material of the cell, and specifically what makes you you. And so your DNA is very unique, and it's not like any other person's at all. Okay, and so if we look at a cell, we'll notice here, this is a non-dividing cell. And so here is our DNA. All of our DNA is pretty much uncoiled. There's no real organization to it, so it's randomly separated. And so that's what we're going to be looking at. Now, what is our DNA made out of? As a review from last class, our DNA is built up of nucleic acids. And so we have each DNA is composed of nucleotides. Nucleotides are composed of 3 different segments. We had a sugar, a phosphate, and then we had a base. Okay. And so with our DNA, here's our phosphate. Our sugar was deoxyribose sugar, because this one didn't have that hydroxyl group. And then we have our bases. Okay. And so here in DNA, A and T, those are always going to come together. And C and G, remember I told you I was always at color guard practice. Alright, uracil, we only find this in RNA. Good. Alright, so ribose, that's also going to be RNA. Deoxyribose, this is DNA. Just look at the first letter. Okay. Let's look at the structure of DNA versus RNA as a quick review one more time. DNA, we have a double-stranded helix with complementary nucleotides. C with G, A, T, C, G, A, T, A, and T. Here is our RNA, and RNA, this is going to be a photocopy of one of the strands of DNA. And we don't want to take DNA out of our cell, and I'll show you why in a little bit, of our nucleus, I mean. And so because it's so long and so huge, we don't want to take it out. We just want to take a photocopy of that. And that's what the role of RNA is going to be. And so here we have that nice double bond, keeping it nice and tight. C and G, nice triple bond. Pairing A, T, or you can have T and A. C, G, or you can have G and C. And that's only when you look at DNA. So looking at these two bonds together. Sugar was at deoxy ribose. Okay, now we're going to go over specifically why DNA is important. And it's important because it contains that blueprint of how we can synthesize and create all the different proteins within our body. And all of this encoding and all of these instructions to create these proteins are found in regions called a gene. Okay, so genes are segments of DNA that carries a blueprint for building one's proteins. And your heredity, so one's heredity is carried by genes. Okay, so here's our nucleus. You're going to notice that this one is actually condensed, and this is because the cell is probably splitting. But basically what we're going to look at are one of these chromosomes. And so in our nucleus, we can organize all of our genetic information into chromosomes. And if we, so here's a chromosome. Here's our DNA. When it's about to, so let me just draw this out for you really quick. Here's our nucleus, here's our DNA, and a cell that's going to not divide yet. When it's preparing to divide, it's going to form nice condensed states called this chromosome, which you see over here on the bottom. But really, what this is, the gene to copy all your DNA, I'm going to make it long, that's just one of these segments over here. So taking a portion of that condensed form and then unwinding it to make it nice and flat. And so here we see that familiar double helix. And if you look at your chromosome, okay, it looks like one hot mess coiled up, but this is all composed of genes. So this gene here can code for eye color. This one can also code for eye color, but maybe let's say this one will code for brown, for those of you who have brown eyes. This one would also code for eye color, but let's say it's blue. Okay, here we can say it's green. And then say hazel or whatever it is. But your true eye color, what you have in your eyes, so I have brown eyes, that's because this gene carrying the instructions for brown eyes is the gene that's being expressed. This is the one that's being copied. None of these other genes, I may have these genes within my body, within my DNA, but they're not being copied down, and therefore I don't have that expression within my eyes. Now, this entire sequence of all these genes is super, super long. We don't want to take this entire segment, this entire DNA, outside of our cell. We only want to copy it, right? I don't need this whole entire line of how many other colors of eye possibilities that I can have. All I want is this gene right over here that'll tell me all the instructions to make brown eye colors. So this is what we're going to be looking at, this process of copying it, copying this instructions, this gene, and then making it into something known of proteins. Okay? So let's clear that off. And so what genes are, they're sequence of 3 nitrogenous bases, meaning those nucleotides. So they're composed of 3 nucleotides that codes for a specific protein. Oops, I'm sorry, I did see that right here. Okay, so we can just put that over there. So sequence of 3 nitrogenous bases that codes for a specific protein. Okay, and so eventually the function of this is to produce your protein. Okay, and so here we're going to take 3 nucleotides. This is going to encode 1 amino acid. Here's the next sequence that's going to encode for another amino acid. These 3 is going to encode for another amino acid. These amino acids come together, and then eventually, remember our last chapter, amino acids, primary structure, eventually it's going to come together, form its 3-dimensional shape, and then form its functional protein. Alright, so RNA, how is this related to DNA? The function of this RNA is that it's going to, oops, I didn't spell it right, function, copy a strand of DNA. Okay. And so it's created from a single template of DNA, contains a sugar ribose, and the base is AUCG. And so if we have, let's say we have DNA, what's our RNA going to be? If our DNA is A, what's RNA going to be? It's going to be U. If we have T, what's our RNA going to be? It's going to be A. If we have a C, we're going to have a G on RNA. And if our DNA is G, we're going to have a C on RNA. All right? So we're making a photocopy of that. And there are 3 different types of RNA. We have messenger RNA. This one's going to do all the dirty work for us. We have transfer RNA. This is going to bring our specific amino acid that we need. And then we have ribosomal RNA. This very first part of how we produce this protein, this is called protein synthesis. And so protein synthesis has 2 parts to it. We have the first part of transcription. Transcription is the act of copying RNA from DNA. And then we have translation, so making sense of that RNA that we just copied into making that final protein and translating that copy into amino acids to form that protein. So let's start here off with protein synthesis. And the first phase of protein synthesis is transcription. So transcription is the production of RNA from a single strand of DNA. And I give you this example of a library and just photocopying one book. And if you think about all of these textbooks, okay, you line them up directly next to each other. Here we have them kind of stacked, right? So you have one row, then you have the other. But instead of having them stacked, if you create one extremely long line of all the textbooks adjacent to each other, you know that all these textbooks don't have the exact same story. They don't have the same contents. One of them can be about history, one of them can be about engineering, about life, about science, psychology, so many different topics, so many different focuses. Okay, and so that itself can be considered just like how your DNA is. Okay, so your DNA is composed of lots of different regions and segments that specify different things. And so one segment, just like one book, can specify your height. Another segment, just like, for example, an encyclopedia or several books, this can specify your liver development. Okay. Another section, this can specify the formation of your hair, what type of hair you're going to have, what color of hair you're going to have. Okay? And now you're not going to take, let's say that now we want to focus on your hair, right? And we want to focus on pigment. You're not going to take all those books from that one section. You only want to take one book that will give us instructions for hair color. And so what do you want to do? You want to just photocopy it. So rather than taking everything out of the library, you're going to photocopy just one book. And just several pages are going to give you the instructions for the pigment of your hair. So you just want to photocopy that. And that way you can take it home and then you can carry out the whatever instructions you need for that hair color. Okay, and so that's exactly what transcription does. It's going to produce RNA from a single strand of that DNA by photocopying a part of that segment. So these are the genes. Okay, so all of these are genes. All right, and so how does that happen? Okay, so here is our gene for that hair color. So we want to create the proteins that'll cause our hair to be a certain color. Let's say we want it to be brown. And so we're going to copy just this segment, just this gene. Here is it flattened out. We're going to start over here, GAG. So we have this protein called RNA polymerase. It's going to help start initiating this transcription of our gene. We don't need to know these different terms, right? But what we do need to know is that these are going to be read in a series of 3 nucleotides. And we give that a term of a triplet. Okay, so here's a triplet. And these triplets, they will encode something. So that's what we give the term a codon. So these triplets, basically that means it's a series of 3 nucleotides. And these triplets form a codon. And what a codon does, it specifies an amino acid. Okay? And so here we're always gonna read in 3. So 3, 3, 3. Okay, so we're gonna start it here. Our G, we're gonna have what? So G is on DNA. Here's our DNA strand. Here's our RNA strand that we're gonna form. G is going to form what? G will be with a C, A will be with a U, G will be with a C. The next, what are we going to have? T is going to be with an A, so good, we see the A. A will be with U, C will be with G, G will be with C, and then G will be with C again. And so we're going to continue until we're done with this gene. Okay, so what you can do right now, take a moment, we'll take a pause really quick, and then go over what your answers would be. Alright, and so after you've paused this video, G with C, G with C, C with G, T will be A, C will be G, G will be with C, T will be with A will be with U, and then C will be with G. Okay, now these will then encode an amino acid, which we do not have to know. However, each of these will be specific. So an AUG, that could mean it could encode one of the amino acids such as glycine or leucine or isoleucine or whatever it is, but they're specific to this codon. Okay. Now the second part of the protein synthesis, this is called translation. And so if here we had the recipe of how to create brown pigmented hair, or we can just say we went to get a Martha Stewart magazine and she has this great recipe and we want to copy it at home, now that we have a copy of it, we can make sense of it. So we can add the ingredients, put something together to form that final product. So translation is just that. It's assembling our protein by reading the codons and adding amino acids using the information carried by the mRNA molecules. Okay, so let's see. Alright, we don't want to take all that DNA, it's too long. Here it is inside of our nucleus, it's too long. We took a photocopy of that gene. Now we photocopied it mRNA, messenger RNA, this is the only thing that can leave. It's small enough to leave outside of our nucleus. We don't want to bring our DNA out. And so now how do we make sense of this messenger RNA to form an amino acids? Here is our codon, remember, composed of those triplets, those triplet nucleotides. And so 3 of these nucleotides forming that codon is going to be paired up with its opposing 3 nucleotides. And so this is a codon. This is from that mRNA. And then here, the opposite to that, the complementary to that, is going to be called the anticodon. Okay? And so here you see the structure. So we had, remember, 3 different types of RNA. Messenger RNA, this is the one we just looked at. It just came out of the nucleus. This is the only thing that can exit the nucleus. From here, we're going to bind to those ribosomes. Okay, so you remember those ribosomes? They're either free-floating within our cytoplasm, or they can be bound towards or on our rough endoplasmic reticulum. Okay, and so here's our second RNA, and this is going to be ribosomal RNA. So that's going to be this right over here. And so all of these ribosomes, their main function is that protein synthesis. Messenger RNA is going to go towards one of these ribosomes and help it decipher whatever is going on. Third type of RNA we have is transfer RNA. So that's where we find these anticodons. Anticodons are going to be complementary to our codon. And on the other end is going to be that amino acid. And so here we have UAC. This is going to encode amino acid number 1. And it'll bring it towards our ribosome if and only if we have this complementary codon and anticodon. Now what's going to happen? We have 2 different units here. Okay, first unit, this first 3 sequences, it's going to bind. You're going to bring in this tRNA. It's going to nice complementary bind. Then we have our first amino acid. Now we're going to read the second codon, CCG. We're going to look into our cytoplasm, cytosol. Is there any other tRNA that has a complementary anticodon to that? Good. So we're looking for something with a GGC since we have CCG. So this is all the complementary. We have something with CCG that's going to come, or GGC. It's going to come here, bind the GC, and now what happens to our amino acids? How do they form a bond? Dehydration synthesis. So we're going to remove a molecule of water. Now we form this bond between 2 amino acids. It's called a peptide bond. Our messenger RNA strand, it's going to move over another 3 nucleotides. And now we can read the next sequence. So we have AGC. What are the next things that we need? A is going to go to U. G is going to go to C. C is going to go to G. Perfect. We found another transfer RNA is going to come in, complementary to that. And then what's going to happen? This is going to come adjacent to amino acid number 2. We form our peptide bond. And we're going to go down this entire list until finally our end of our messenger RNA is done. Now we have our primary structure. So primary protein structure is all it is. It's just a sequence of amino acids. And then now we can start refolding it to form a 3-dimensional conformation. Okay, so make sure you can go through this and hopefully it'll make sense for you. Now let's review everything again step by step. We have here our DNA. We undergo transcription or translation. Good. Transcription, we are transcribing, we're writing something down, copying it. We can't bring the entire DNA out of the nucleus, so we're going to bring our messenger RNA out. It's going to find one of the free-floating or a ribosome bound towards on our rough endoplasmic reticulum. Then we're going to start reading our sequences. So the first 3, here's our codon. This is called what again? So anticodon. This is the complementary sequence of nucleotides to the codon. Alright, and then of course, what's attached on the other end of this? We have the amino acids. And so here, these three is going to bring in this amino acid. UUC, complementary, was going to be AAG. It just left. That encoded for phenylalanine. Okay. These encoded for serine. These encoded for glycine. And so it's going to continue to create its strand, and then eventually we'll have our conformation for our protein. Alright, so it's going to move along 3 at a time, and then finally release its tRNA once it's done binding its amino acid. This is a great video to watch for you to go over transcription and translation again. So anytime I post these videos, it's a great thing to watch just because it helps you clarify some concepts. Now here are those proteins being synthesized. Here's a messenger RNA, ribosome. It's getting synthesized. Here's one that's bound on our rough endoplasmic reticulum. And as it synthesizes those proteins, it's going to migrate into the rough endoplasmic reticulum cistern. So cistern is just kind of like this cavity or hole or region in the middle that's open where we start collecting, gathering our proteins. Now here we're going to fold into the final functional shape. We can then package these into sacs called transport vesicles. And as a review, where are these vesicles going to go? Good. So it's going to transport vesicles. We're going to bud from our rough endoplasmic reticulum and travel to the Golgi apparatus for further processing. Now here, Golgi apparatus receives all those proteins from our rough ER. Oops, sorry. And it'll package these proteins. So here's a rough ER. We have all of those newly synthesized proteins inside there. And they're going to package them to vesicles destined for one of 3 places. It's going to form a lysosome. Number 2, it can eventually excrete contents into extracellular space, meaning that it can secrete it outside of the cell. And then finally, it can regenerate the cell membrane. So we can create more of these proteins of our cell membrane, or the vesicle itself can fuse to become part of the cell membrane. All right. Now, a lot of times, exocytosis, where will this happen? Well, you think about your sweat glands. Your sweat glands are lots of oil. I'm sorry, not sweat glands. I'm thinking of your sebaceous glands, your oil glands. And so anytime you have an oily face, you're going to secrete a lot of this contents from that. We'll smooth ER, since we create a lot of those lipids. And you can excrete it, and that'll come out. So when you get an oily face, that's really this secretory vesicles releasing all of that oil into that extracellular space, which will then get secreted out of your body. Now here is a review of those 3 pathways again. Here we have exocytosis. Proteins are going to come out. Also oil is going to come out. Here we have that vesicle. We can imagine here having proteins, and this is going to be part of our cell membrane. And then finally we can create more lysosomes. So we have lots of digestive enzymes here within our lysosome. Fuse it. We can break down other ingested material, or we can also just form more lysosomes itself. Great. Now the very last concept we're going to go over is the cell life cycle. And so your cells have 2 major periods. You have interphase, and this deals with cell growth. Oops. And their duplication of your DNA. And so really, interphase is the longest phase. What you're doing is you're duplicating all the proteins, all the organelles within your cell, in order to split and divide your cell in half. Okay, so cell dividing them in half equally. If you have equal division of cells, this is called mitosis. So it's the nuclear division of somatic cells. And somatic cells, those are the cells that are every single cell within your body. These do not undergo reproduction. And so the ones for reproduction are meiosis. And so this is nuclear division of sex cells. Okay? And so the only examples for this we have are sperm and the ovum. Here are the somatic cells. You have skin, hair, liver, etc. Okay. Now, mitosis, a lot of times you're going to have increased rates of mitosis or increased rates of cell division, specifically for cells that continually have to regenerate and replace themselves. And so your skin cells, your hair cells, those are constantly having to become regenerated. So they have a higher mitotic rate than something like your skeletal muscle cells, which will only undergo division one time. And then once you destroy or damage those types of cells, they can never regenerate themselves once more. Okay. Now, apoptosis, this is called programmed cell death. In the event that your DNA ever gets damaged, automatically the cell is going to undergo apoptosis. However, if your cell is damaged and if your DNA is damaged and your cell does not repair it, that could eventually lead to something like cancer. So you're continuously repairing damaged DNA, but those DNA itself is not encoding the correct genes, not encoding the correct proteins. And that's how you can result in disease. So looking at the cell life cycle, mitosis is a separation of duplicated chromosomes into two identical daughter cells. Okay, identical because it's this exact genetic material. Here's the DNA. You have one from your mom, one from your dad, one copy of genetic information each. It's going to duplicate, so here's one, here's a copied area. And then here, mitosis, what we have to do is separate this duplicated material into two daughter cells. And so here, exact copies going to one side, exact copies going to the other side. Now we have two identical daughter cells with the exact same genetic information. Okay, and so really we're going to focus on for the next couple of slides is the somatic cell life cycle. And so this is split into two major phases, interphase and mitosis. Interphase, we looked at, prepares the cell for growth and DNA replication. And this is so long because, as you can imagine, we really need to ensure that all of the organelles, proteins, and DNA is correctly duplicated in order for it to have, in order for no disease or anything to occur in the next cell life, okay, the next cells. All right, and then the short phase we have is mitosis. And so mitosis has a series of a couple of phases, and this is a process of physically separating the two nuclei. Okay. You'll often see different terminology in your textbook: chromatin, chromosome, and sister chromatids. Let's first look at a non-dividing cell. We looked at this earlier. Here you have your chromatin, your genetic information in the form of DNA. But if you notice, it's not very organized itself. It looks kind of like free-floating noodles. And so this is when you call DNA chromatin. Okay, so it's in a non-dividing cell. Now, when the cell prepares for division, you're going to have, here's one chromosome, here's another chromosome, but what's going to happen is that it's going to start condensing itself. Okay, and here is going to be the duplicated material. All right, so that's why you see two, but really they're bound together. So what you're going to see is a lot of times the DNA looking just like that. Here I'm showing you two separate ones just for the sake of showing it to you. So this is an exact copy of this one right over here. And the whole idea is we're going to separate it into its two daughter cells. So this is supercoiled. It's called chromosome because it's clearly visible. And then finally we have what are called sister chromatids. And so here, because these are, well, collectively this is one chromosome. So here's one chromosome. And here, each half is called a sister chromatid. Okay, so chromosome being that entire unit, and then chromatid being one individual side. Right. Now, in the mitosis, it's this process separating our nucleus into forming two daughter cells, and specifically also our other organelles. We undergo 4 phases, and I remember this by the acronym PMAP. So you have prophase, metaphase, anaphase, and then telophase. Okay. And let's go over what do these entail. So here we have interphase. Interphase, we have duplicated genetic material with duplicated centrioles. And now we're going to undergo early prophase. And so during this prophase, what happens? So really early, late prophase, a couple of things happen. Number 1, the nuclear envelope breaks down. Number 2, if you look at your DNA condenses to form chromosomes from chromatin. And then number 3, you have the formation of microtubules. And so microtubules, this is going to assist in the movement of all of these chromosomes. So we want to make sure that we can have all our chromosomes lined up for that, and that's what exactly those microtubules are going to do. Now here, the next phase we have is metaphase, and metaphase deals with aligning the chromosomes along the metaphase plate. Okay, here is this imaginary line cutting this entire cell into half, and so here we're going to start aligning and arranging our DNA to be nice and flat. So then eventually what's going to happen, here we have microtubules. The function of this is once more to help separate all of the organelles and then also separate our DNA. So this microtubule is going to attach on one side of the sister chromatid. The other one is going to attach to the other side of the sister chromatid. And this is what metaphase is. So metaphase is aligning all of these chromosomes along this midline, and also attaching these microtubules to the center of that. Now the next phase is going to be anaphase, and anaphase deals with pulling the sister chromatids to opposite poles. So this is called the pole, which are the centrioles. So those centrioles. And the way I remember this is because as they get pulled, they kind of resemble the letter A. And so A, I think of anaphase. M is for middle, because they're all in the middle line. Okay? And another one that you need to remember is telophase and also cytokinesis. And so telophase, what this does, this is the exact opposite of prophase. And why is that? Well, here we have the nuclear envelope reforming. That's number 1. Number 2, this DNA is going to start decondensing, meaning it's going to become its nice hot mess state again. And then finally, the last thing we have is a breakdown of these microtubules. So microtubules needed to help align and also pull things apart, and now we don't need it anymore, so it goes back towards its centrioles. Okay? And then finally, what's not part of mitosis, but it concludes mitosis, is cytokinesis, and this is the actual physical separation of these two cells into forming two daughter cells. Here's a great video on mitosis. Definitely watch on your own time. Now, looking at the cell life cycle, if that DNA, okay, so we're going to go a little step back. If that DNA is damaged and it's not repaired, what's going to happen is that cell is going to undergo cell death. But if that DNA is damaged and the cell is continuously trying to repair it, what this can lead to is cancer. So cancer, by definition, is uncontrolled cellular growth. Okay, and so looking at how the cells will grow and divide gives you a better understanding of how cancer can arise. And so here, if we don't have any signals that tell our cells to not continue to grow and divide, what can happen? Well, here we have what's a benign tumor. Benign because this cancer cells has not yet entered our bloodstream. The moment that these red blood cells, the moment that these cancer cells enter into our bloodstream, they can spread to other tissues and cells within our body. And once that happens, that's when this is called metastatic. Metastatic cancer, because its ability to spread. Versus this one, this is benign because it has not breached into what's called the basement membrane and has not entered into our blood supply as of yet. Now, it's pretty cool stuff. I did a lot of research looking at oral cancer, and if you know anybody who had some type of cancer, let's say if the person had breast cancer, and why is it that cancer spreads? Well, and how do you know it's actually derived from breast cancer? Each of these cells are unique, so they're going to be destined to be whatever cell type they are going to be. And so cancers, breast cells are always going to be breast cells, and hair cells are always going to be hair cells. And so with breast cancer, what happens is that here, if we imagine this is a cross-section of breast tissue, if this is breast cancer cells, it's always going to have the identity of breast cancer cells. And once it enters into your bloodstream, it can be found into other regions of the body. If it enters into your lungs, it can also become cancerous, enter into another area or site, continue to grow and divide, and then also in turn mess up that organ. And so these cells, because it came from your breast, for example, these are also going to have breast cancer cells. So this is called a secondary cancer because it's able to migrate, and this is a primary cancer. Okay, so that's how that works. Now, I want you to go over these multiple choice questions on your own, and in the next chapter, we're going to go over these answers.